Hey bio team. So if a cell wants to divide, the first thing it needs to do is it needs to replicate its DNA, make a new copy of DNA for the new cell. And so there's a couple of steps to this process of replicating DNA. Here you can see we've got a double-stranded DNA with one strand running from 3' to 5' end, and the other strand running anti-parallel. And in order to replicate this DNA, the first thing the cell needs is a couple of enzymes. This circular enzyme here is topoisomerase, which is an enzyme that just helps uncoil this DNA. And the second enzyme, helicase, which breaks apart the hydrogen bonds between the nitrogen bases in the DNA. So topoisomerase flattens the DNA out, and helicase breaks it apart. And so we'll watch these two enzymes unzip this DNA. And after the DNA has been uncoiled, there's now enough space for a third enzyme, DNA polymerase, to come through and build a complementary strand across from the template strand. Now you'll notice that the polymerase reads the template strand from the 3' to the 5' end, but this complementary strand got built anti-parallel from 5' to 3'. And down below on this opposite strand, another DNA polymerase will do the exact same thing. It will read the template strand from the 3' end to the 5' end, and it will build this complementary strand from its 5' end to its 3' end. Now at this point, the helicase and the topoisomerase can keep unwinding the chromosome. And this DNA polymerase will just follow behind the helicase, building a new strand. However, this DNA polymerase is heading off in the wrong direction. So what our cells need is a new DNA polymerase to begin the replication process again at this freshly exposed area on the template strand. And so we'll watch that happen here. And now what you'll notice is that we actually have two fragments of DNA on this lower strand. And these are called Okazaki fragments, named after the married couple that discovered them. But in order to complete the process of replication, we need another enzyme called ligase to attach these two fragments. And so we can replay it. Now, because this strand gets synthesized a lot faster, it's a lot quicker process, this is called the leading strand. And this lower strand here, because it takes longer to synthesize, is called the lagging strand. And that's it. At this point, you guys have some practice problems. We'll see you next class.